Okay, so first we're going to cover histology. And as usual, I do have my histology packet open, and I will cover all of the um, questions, but I'm not going to write out the answers. That is still your responsibility. And if you're not with us in lab right now and you're listening to the recording, you're going to have to fill in the answers based on my discussion today. Believe it or not, I am trying to train you all to be independent thinkers and learners, especially as you move forward from microbiology and physiology. There's considerably less hand holding. Okay, so first slide, and of course, the slide that follows the picture, the histological specimen picture, has the questions that go with the preceding slide. For example, this is our first slide, and the second slide has these questions that go with the first slide. If you actually download the PDF document, it has the histological specimen and the question slide all on one page. And you might like that better to fill out and then email back to me. And I'll remind you that your histology is due on Monday. Okay, so let's get back to this slide. This slide is actually showing you the trifecta. Arteries, nerves, and veins travel together. Oftentimes, all three are wrapped up in connective tissue. So what you are seeing at the end of this dotted pointer here, at the end of this pointer, you're actually seeing a nerve. In fact, you can see the epineurium, and you can actually see all of these fascicles wrapped around with perineurium. So all of this here is a nerve. A whole bunch of axons are coming out at you, if they were 3D, they'd be coming towards your face. Now, the two dashed pointers that are shown here, both of them are pointing to the same type of vessel. Specifically, they're pointing to a vein. Now, how did I know they were pointing to a vein? Veins don't have thick tunica medias. And as such, they are prone to collapse. And that's why these veins look like they're not popped open as well. They look flat, sort of like a deflated balloon. So again, how did I know these were veins? Because I could see the tunica intima kind of all bunched up because these veins have collapsed. They don't have good structural support from the tunica media. In contrast, the solid pointer is pointing to the thick tunica media of an artery. And in fact, you can see some blood cells still remaining in this histological specimen. So again, at the end of the solid pointer, I can see it's an artery, and I know this based on the thick tunica media. So all of this here is the tunica media. And again, that gives a lot of structural support to keep that blood vessel patent or open, not collapsed like the veins appear in these pictures.
So wait a minute, Professor Street, the dotted arrows, they're also tunica media as well, like you said?
Well, the dotted or the dashed?
Here, hang on. Sort of like the dash, but they're disconnected.
So
Not the
dash.
dot.
You mean the ones circled in purple?
Yes.
Those are dashes. So those arrows, the arrows specifically are pointing to the vessel. So the dashed arrows are pointing to veins.
Okay.
guess if you're asking what is the arrowhead landing on, yes, the arrowhead is landing on the tunica media for
Okay.
those two dashed specimens.
Okay. Thank you.
You're welcome. In picture 3, we are seeing an artery and a vein next to each other. And I'll leave it to you to figure out which one's the artery and which one's the vein. I'll give you a hint. Again, it's the thickness of the tunica media. Now, the arrow in this picture is actually pointing to connective tissue that surrounds many arteries, veins, and even nerves when they're wrapped up with them. And this connective tissue looks like it's just air bubbles, but in fact, you're looking at adipose tissue. And every single one of these circles is an actual adipocyte. And it doesn't look like they have nuclei, but their nuclei, if you look right here, the nucleus is actually smushed towards the edge of the cell. So every once in a while you see these little black dots towards the cell membrane, and it looks like it's an artifact of the specimen being preserved, but it's not. Those are actual nuclei shoved up just underneath the plasma lemma, underneath the cell membrane. Okay, how are we doing so far? Does everyone remember the 3 tunics of blood vessels?
Yes.
Okay, most blood vessels have 3 tunics, but remember capillaries do not have all 3 tunics. They simply have a tunica intima with a basement membrane and the pericytes, support cells that help stabilize them. But in this picture, the artery and vein, both of them have 3 tunics. There's a tunica intima, a tunica media, which is very thick in arteries, and a tunica externa. Okay, now slide 5. Slide 5 is showing you a portion. No, strike that. Let me rephrase this. Slide 5 is actually showing you, might help if I get on slide 5. Come on. Click on, there we go. Slide 5 is showing you a longitudinal section through an elastic artery, like the aorta. And when we think about elastic arteries, we know one of their amazing features is their abundance of elastic tissue. However, the elastic tissue is not arranged in sheets, also known as lamina. There is no internal and external elastic lamina in elastic arteries. Instead, their elastic tissue is found threaded within the tunica media. And in fact, that's what you're seeing. This entire slide is showing you a section through the tunica media of the aorta. And all of these red, squiggly, pasta-looking lines are elastin threads. And the cells that you're seeing in between are spindle-shaped, and they are mononucleated. Spindle-shaped and mononucleated. So those are the smooth muscle cells of the tunica media. All right, so this could have been, it also could have been a histological specimen of the pulmonary artery. The pulmonary artery, of course, being the elastic artery that comes off of the right ventricle. So either the aorta or the pulmonary artery, those are 2 very good examples of elastic arteries that have elastin threads interspersed within their tunica media among the smooth muscle cells. Now we're moving on to our formed elements. Now, there are a few formed elements shown here. First of all, there are no arrows pointing to these, but this is your most numerous formed element and also your most numerous cell type in your body. You have about 200 different cell types, and 1/4 of you is from these red blood cells. So 1/4 of you are 100 trillion cells big, and 1/4 of you consists of red blood cells, truly your most abundant cell type in your body. These little fragments that you're seeing here, those little fragments are platelets. Remember, platelets are pieces of a megakaryocyte. Now, so here again is a red blood cell at the end of that black pointer. We know our white blood cells can be grouped into either granulocytes or agranulocytes. Both of these cells at the end of the yellow pointer and red pointer are granulocytes. They are fills. Remember, our fills are filled with granules. So at the end of the red arrow, we see an eosinophil. And we know this because we can see all of those red granules, a whole bunch of them. They like to bind the acidic dye called eosin. And that's why they stain kind of a pink reddish color. The nucleus here is a polymorphonuclear nucleus, which means it's it's just one nucleus, but it looks like we've got a lot of different like multiple nuclei squished together. We do not. It's one nucleus, and it's sort of like an animal balloon that's been shaped. So we can see this eosinophil has kind of an S-shaped nucleus to it. Eosinophils mature into that is not true. I almost lost myself. Eosinophils are important to fight parasites, specifically worms that might be found in our gut. So eosinophils are very good for that. Eosinophils are also very good with helping to end allergic reactions and inflammation. They help end it. So I think E for eosinophil and E for end to help me remember that they are important for stopping an inflammatory process. The other fill that you are seeing at the end of the yellow pointer is a neutrophil. It is your most abundant white blood cell. It is a non-specific type of immunity. Neutrophils are very good phagocytic cells. They're not as good as mature monocytes that turn into macrophages, of course, but they are still good phagocytic cells. And again, they are non-specific. They will eat up dead or dying cells. They are good at phagocytizing bacteria, cellular debris, etc. And I hope everyone knows what an erythrocyte or red blood cell does for your body. I hope we haven't forgotten what we learned in lecture unit 2. And here is another picture of your red blood cells. Notice that these red blood cells, this is just kind of an interesting, they look spiny, don't they? And that's because whoever prepared this slide did not use the best solution to keep those red blood cells happy. They have kind of shriveled, they've kind of crenated, and that's why they look like their cell membrane is a little bit blebby and then they're not looking perfectly round. So the isotonic saline solution that they were put in clearly was not prepared very well. At the end of the green pointer, we see the same cells that we saw on slide 7 at the end of the yellow pointer. So again, those are neutrophils. And at the end of the black arrow on the right of your screen, we are seeing a granulocyte. It is not a fill, and it has this U-shaped polymorphonuclear nucleus that takes up almost all of its cytoplasm. And this A granulocyte is a very powerful phagocytic cell. And it is usually it leaves the blood vessel and goes into tissue beds and matures into another name. It matures into, I shouldn't say it matures into another name. It matures and then we give it a different name. So does anybody know what cell is at the end of the solid pointer?
Is it monocyte?
It is a monocyte and it matures into a macrophage. Now this cell is part of your specific immunity, and it does not have a polymorphonuclear nucleus. The nucleus is round and occupies almost the entire space of the cell. This is a lymphocyte. There is no way for you to look at this lymphocyte and figure out if it's a T cell or a B cell. So when you write down in your histology packet, what does this cell do for the body? You should state that it is part of your specific immune system. And then you should state what a T cell would do and what a B cell would do. So a B cell has to mature into a plasma cell, and you should state what that cell would then do for your immune system. There are different kinds of T cells that you learned about in lecture unit 2. For your histology packet, I would write down what a cytotoxic T cell does. I wouldn't worry about helper T cells or killer T cells. I would describe what a cytotoxic T cell does. And again, you studied this back in lecture unit 2. So I'm hoping the memory recall is not too difficult for you. We are quickly approaching final exams. I know you're not thinking of it, but I'm already thinking of it. I mean, we are in week 12 and there are only 16 and a half weeks in this semester. So we're almost there. We have about a month left. I can't even believe it. And it's going to go by so quickly. So now we're on slide 13. And what you're looking at is actually an up close, a more higher magnification of the same structure you looked at back on our first slide, up here circled in yellow. So you are looking at a nerve, real up close, high magnification. All of these dark circles in the middle, those are actual axons. And notice there are different sizes of them. There are some neurons shown here with large axons and some with very small axons. Does size matter for an axon? Absolutely, absolutely. The bigger the diameter of an axon, the faster the action potential can spread or travel down the axon, the faster it can travel. The smaller the diameter of an axon means that neuron will have more resistance. It's smaller. It's a lot like, have you ever been traveling down a 4-lane freeway, let's say, and it's late at night? This used to happen to me all the time. I teach night classes at Saddleback. And I usually choose night classes because the commute from where I live in Huntington Beach to Saddleback, Mission Viejo, that should be a 35-minute drive door to door. But it frequently is not. So I liked teaching night classes, and I would show up around 3 o'clock in the afternoon for office hours, even though I didn't teach until like 5 or 6 o'clock p.m. So I'd show up a little early so that I wouldn't have to sit in traffic. And then when classes were over, it was a breeze to get home most of the time. That is until about 2 years ago when they started redoing the 405 freeway up near Fountain Valley and Huntington Beach. They're redoing all the bridges and they're redoing the carpool lane. I don't know if you know this. But before COVID hit, and I'd be driving home at 10:30 at night after class, thinking I'd be able to zip home and not hit any traffic, all of a sudden, of course, they do construction on freeways at night, don't they? So all of a sudden I'd be zipping along, and then I start seeing the orange cones starting to move everyone over down to 1 lane. And if everyone is, even if it's 10:30 at night and everyone's down to 1 lane in what should be a 4-lane freeway, you're going to hit resistance, you're going to hit traffic. And it still would take me about an hour to get home. I wouldn't get home till close to 11:30 at night. It was just craziness. So I hate to say it, but it's one of the things I'm thankful for COVID-19. My commute is so easy compared to what it was. Heck, if I want to save commute time these days, I just sleep on the couch and then I just roll over and there's my laptop. It's brilliant. Anyway, axons are a lot the same way. The bigger the axon, think 4-lane freeway, traffic can zip along a lot easier. There's less resistance. But if it's a small axon, there's more resistance, and then the action potential does not travel as quickly down the axon. So there's a question on your histology packet that asks, do you think size matters? And the answer is yes, it does. Now this space that you're seeing in between this is actually a fascicle. So there's fascicle 1. Here's another part of another fascicle. That's fascicle 2. And here's another part of a fascicle. So that'd be fascicle 3. So this space in between here would be the perineurium. Just put perineurium. So overall, we are looking at a collection of axons in the peripheral nervous system called a nerve. All right, now this is a transverse section through a spinal cord. And spinal cord, the question asks you, what kind of tissue is this? You should be able to tell me the 4 tissue types from chapter number 4. There are only 4 basic tissue types. And the brain and spinal cord and all the neurons that you've been studying and all the nerves that you've been studying, of course, it's nervous tissue. So what kind of tissue is it? Nervous tissue. There is a meninge that the green arrow is pointing to. Remember, we have dura mater, we have arachnoid, and we have pia mater. And pia mater is tightly adherent to the surface of the brain and spinal cord. And dura mater means tough mother. Pia mater means gentle mother. So the green arrow is showing you dura mater. Now, coming out of the spinal cord, exiting our ventral roots. So in yellow, you're just seeing slices through the ventral root. And then that means up here, this slice here, and over here, it's showing you the dorsal roots that are coming into the spinal cord. And if you give me just a moment, I'll show you another picture of a spinal cord where you can actually see the dorsal and ventral roots coming into and out of the spinal cord. So I'm just going to pull up a different histological specimen for you. Just give me a moment. I'm going to unshare the screen and then I will re-share it for you. As soon as I find the one that I had pulled up. There it is. Found it. Here it is. So over here, this picture that you can now see in better detail on the right side of your screen, you can see the dorsal root coming up here. Let me just grab that. Copy. I'll grab it and then I'll put it on another slide just for demonstration. Just for demonstration. Okay, now I can draw on it and show you. So again, we can see the dorsal root coming in here. And here's another dorsal root coming in. And then we can see the ventral root leaving. And down here, another ventral root. And in our picture, we can still see in green that I've highlighted around the dorsal roots and in yellow, the ventral roots. It's just, why do they look circular instead of stringy like the slide that I just pasted? It's just because of how the machine that was slicing through the specimen was cutting through. I mean, if it was cutting through the spinal cord and eventually slicing its way out of those dorsal and ventral rootlets, we would only be seeing little fragments of them. So that's why they look circular, and you're looking at that going, that doesn't look like a ventral root like this slide does. Again, if you start slicing through it and you're getting through the entire thickness of the dorsal or ventral root, eventually you just have remnants of it left. So if you don't believe me, you're just going to have to take my word for it for testing purposes. And then at this point in the semester, especially after taking your Lecture 3 exam, you should be able to tell me what white matter is and what gray matter is. This is a picture of a multipolar neuron. So it is nervous tissue. Of course, it is a neuron. We can see its nucleus in the cell body. And the cell body is also known as the soma or perikaryon. And we can see its axon coming off of the cell body. And the green arrow is showing you the beginning of the axon. And the beginning of the axon, the part of the axon that communicates to the cell body or attaches, comes off of the cell body is a better way of saying it, has a special name. And it's called the axon hillock. And you should know from both lecture exam 1 as well as lecture exam 3 that at the axon hillock, this is where action potentials first begin. Why do action potentials first begin here? And it's because we find special types of channels that open because of voltage changes, like sodium voltage gated channels and potassium voltage gated channels. Here is another picture of multipolar neurons. Specifically, this came from, they're not human, it came from ox. So they're oxen multipolar neurons, not that that matters. Ours would look very similar to this. And all of those tiny little dots that you're seeing around these multipolar cells, these multipolar neurons, are support cells. And we learned from lecture unit 3 that there are 4 types of support cells in the central nervous system and 2 types of support cells in the peripheral nervous system. So these little dots, we don't ask you to tell us, is it microglia or is it astrocytes? We don't ask you to identify them. Just know that those dots are some of those supporting cells that you learned about in lecture unit 3. Okay. So where can you find neurons? I don't think I need to really answer that question for you. Where in the body do you find neurons? And if you still need help with that after lecture unit 3, probably you will want to go back and read your chapter on the introduction to the nervous system. It says give a big list. You can start out with the big 2, like the brain and spinal cord. Those are not the only places where you can find neurons. Okay, now we're moving to another structure. This is actually a picture of the eye. And this colored part here is part of the vascular tunic. That is the iris. This space here in between would be the pupil. And the pink disc looking structure that you're seeing here is the lens. And we can actually see a space behind the iris, but in front of the lens, and a space in front of the iris, but would be behind the cornea. And the cornea is not shown here, but I'm just going to draw it with a green pen. Just let's pretend it's there. And then this dotted line back here is showing you a space behind the lens, which would of course be the posterior cavity, which is filled with vitreous humor. So posterior cavity, posterior cavity, and then we have the anterior cavity. And the anterior cavity, I'll remind you, is divided into 2 smaller areas. And we're going to see another slide where this is asked of you to identify all of these different spaces. Okay. So remember, anterior cavity can also be called anterior segment, and posterior cavity can also be called posterior segment. Now this picture is showing you the retina. And we know that there are different layers of the retina. Down here specifically, this layer is not part of the retina. Instead, it's part of the vascular tunic. This is the choroid. And the purpose of the choroid, I'll remind you, is to prevent scattering of photons. We don't want them to escape. We want them to interact with the actual photoreceptors. There are two kinds. There are cones and there are rods. So at the end of the dotted line are the photoreceptors. So all of this area here would be the photoreceptors. Now this part, it's not tagged or anything, but students oftentimes say, what's this tiny little area that seems to be all black? That's the pigmented layer of your retina. And you read about that also working a lot like the choroid, where it prevents scatter of photons. And then we have this layer. This layer are the bipolar cells. So I'll just put BP. And then where the dashed line is showing you are the ganglia. And I'll remind you, all of their axons travel out to the optic disc. And then all of those axons exit the eye as the optic nerve. A question asks, how does light travel through these layers? And light travels by going past the ganglion cells, past the bipolar cells, hits basically the choroid and the pigmented layer, past the photoreceptors, and then ricochets back and then interacts with the photoreceptors. They get excited, they start the action potential, and then the action potential goes towards the bipolar cells, then towards the ganglion cells, and then out through those axons into the optic nerve, which is cranial nerve number 2. Sorry, I'm just trying to abbreviate. Now we only have 2 more slides for the histology. This is another picture of the eye. And let's give this person green eyes. So I'm coloring in part of the vascular tunic that is pigmented. That would be the iris. This structure here is the ciliary body. I'm just going to put CB. And these little pieces that are coming off here would be those ligaments that anchor into the lens. So here's the lens. And this part going around the anterior surface of the eye would be the cornea. And this space in front of the iris, but behind the cornea, would be the anterior chamber, that's a C, of the anterior cavity. And of course, it's filled with aqueous humor. And the dashed dot, dash dot line here is showing you the posterior chamber of the anterior cavity or segment. And that is behind the iris, but in front of the lens. And then already shown on another slide, of course, this would be the posterior cavity filled with vitreous humor. Now back here, the cornea is continuous. The cornea, I'll remind you, is transparent to let light through. But it's continuous. It's part of the fibrous tunic. It's continuous with the white of the eye. So this dotted line is showing you the sclera, or the white of the eye. And then lastly, our last slide is a histological picture of one of the ear models I showed you a couple of weeks ago, where we saw the scala vestibuli and the scala media. The scala vestibuli is filled with perilymph, just to remind you. we're trying to put together lecture material with lab material. Remember, if you're studying with one, for one, you're studying for the other one as well. The scala tympani is also filled with perilymph. And the scala media is that soft membranous structure within the cochlea. So scala media can also be called the cochlear duct. The black arrow is showing you the vestibular membrane. And the cochlear duct, of course, would be filled with endolymph. The green arrow is showing you the basilar membrane. And this entire structure that I'm going to circle in green is your organ of Corti with the little hair cells. And those hair cells are going to, once the basilar membrane starts undulating, starts flexing up and down from the fluid waves created from the pressure waves when the ossicles start moving, those hair cells get smashed onto the tectorial membrane. The tectorial membrane should not be confused with the tympanic membrane. That's the tympanic membrane separates your outer ear from your middle ear. And all of this is showing you your inner ear. Okay, are there any questions regarding the histology?
I have a quick question for slide 25.
For what? I'm sorry?
Slide 25. I wanted to know, like, what angle do you look at that? Like,
What do you mean, what angle?
like, okay, because you're looking at the color of the eyes, but I feel like,
Are you talking about this slide?
no, 25.
Well, okay, so
Right.
20.
They're
Okay,
different for you.
hold on. I inserted a slide, so let me take it out. So this slide.
Exactly. Yeah.
Okay. And your question is again, I'm sorry.
Like, how do you look at that? Like, what angle is that of the eye? Because I know that the color of eyes is right there, but like, I kind of don't get the layout of it. If that makes sense.
Okay, this could either be a superior view that you're seeing, looking down at someone's eye that's been sliced from an anterior to posterior perspective, or you could be looking up from underneath, looking up at someone's eye that's been sliced from an anterior to posterior.
Okay, thank you.
Does that work?
Yeah.
Okay. Yeah, so I think it's easier saying you're looking down and someone's eye has been sliced in a transverse section. I think I have an eye here in my room, but not one that's easily reached. Anyway, it's a transverse section and we're looking down. So the cornea, of course, would be the front of our eye. And then posterior to that would be the iris. And then posterior to that would be the lens.
Okay, thank you. I don't know why I was mixing it up. Thank you.
You're welcome. All right, well then, why don't you take a 10-minute break? I'll see you at 5 o'clock. And I will go over the rest of, the parts of the hit list that I feel like I still need to either review, like the parts of the heart, which should be review, and then show you some parts of the nerves, and then address Lauren's question about, do we need to know their functions? And the short answer is, from this lab exam on through the fourth lab exam, you should be ready to ask or answer more concept questions like, what does this structure do? So yes, do be ready for those types of questions. What does it do? In the case of the cranial nerves, what does it innervate? So for example, cranial nerve 8, you actually saw models of cranial nerve 8, and it's the vestibular cochlear nerve. Well, what does it do? It transmits action potentials that will ultimately allow your temporal lobe to determine what you're hearing, right? So having an idea of what those cranial nerves do and what some of these structures do, there's quite a few of those types of questions on lab exam 3, and lab exam 4 even more of those. But as I go through the nerves when we return from our break, I will give you some pointers. Okay, I'll see you at 5 o'clock. Most of this PowerPoint is actually already posted on under our lab 3 unit in Canvas. However, it was called Vessels and Heart. And because I have redone the vessels PowerPoint, and we did that separately, we did it as arteries on Monday, and last week we did the veins. I have redone this heart and vessels to be heart and nerves. So all of the pictures from the original heart and vessels PowerPoint, all of the pictures for the heart is here. They're the same pictures. And then I've just added a few more nerves pictures towards the end. So I will upload this to YouTube. Sorry, I will upload this PowerPoint as a file under Lab Unit 3. But I assure you, most of these pictures you've already had in the previous PowerPoint that's been there. And all I did was take out the vessels part because there were a lot of cat vessels there. So here we go. This is new and updated for fall 2020. I don't feel like I need to go over too many of the models that are already labeled for you. But there is a structure or two on this picture that is not labeled that is on your hit list. Most notably, this part here is the aortic arch. And specifically where this arrow is pointing to, we should call that the ascending aorta. And then where the aorta is actually going down there, that would be the thoracic descending aorta. We see blood vessels for the coronary circulation already labeled here for the most part. We do see a little tiny bit of the left coronary artery, this part right here that I'll highlight in yellow, and then I'm going to erase it so you can see the red artery. And it branches into the anterior interventricular artery. And as soon as that anterior interventricular artery branches off, then it turns into the circumflex. The blue here is the great cardiac vein. And yes, this picture identifies the right coronary artery, but this artery down here on the side of the heart is the marginal artery. And this vein that is not identified is the small cardiac vein. So those were just a few structures that I thought should be pointed out and identified. Here is another model of the heart, and I won't tag all the blood vessels because I just went through those on the previous slide. But here is the left atrium. And here is the right atrium. Here is the interventricular septum. Septum means wall. So it's the wall in between the two ventricles. This space would be the right ventricle. And this space would be the left ventricle. Take care reading your questions, because if we say, name this chamber, of course you would have said right ventricle or left ventricle, like I just identified here. But let's say you have an arrow pointing here and it says, name this structure. Well, then you would say papillary muscle. And here, of course, is the right AV valve, also known as the tricuspid. And these strings that you are seeing, and this is the left AV valve, also known as the bicuspid and mitral valve. And yes, I would know all of them. Mitral. Those strings that you're seeing are the chordae tendineae. And this bumpy appearance of the muscle down here is the trabeculae carneae. Something like that. Let's see, up here I can see the pulmonary semilunar valve, and way up here is the aortic semilunar valve. Dang it. And here is a heart all opened up, and again showing you a lot of these structures that I just went through. Again, chordae tendineae. Those are the strings. The papillary muscles are the muscles that are, let me get rid of that. It's all filled in. Oh, can't get rid of that. It's already saved. Anyway, the papillary muscles are those finger-like muscles that pull on the chordae tendineae, and shown here on the side with my blue highlight, I'm trying to show you the trabeculae muscles. Oh, here they are, trabeculae carneae. And we find that not only in the right ventricle, but also in the left ventricle. Let's see. These striated muscles here that I have highlighted in blue, and now I'm going over it in purple, are the pectinate muscles. Those we find in the atria of the heart, both the right atrium and the left. Here is a picture of the back side of the heart, and we can again see some of the blood vessels for the coronary circulation. We see the coronary sinus, of course. And on the back, this red vessel here is the posterior interventricular artery. And traveling with it is the middle cardiac vein. We can see the entrance of the inferior vena cava into the right atrium. We can see, excuse me, the paired right And left, we're only seeing one. The other one would be more anterior there. Pulmonary veins, they are bringing blood back from the pulmonary circuit, oxygen-rich blood, into the left atrium. And again, we can see the descending thoracic aorta. Just a little bit of fun to identify the different structures here. Number one would be showing you the aortic arch. Number two would be showing you the remnant of a fetal structure, the ligamentum arteriosum, which is the adult structure for the ductus arteriosus. Number three is showing you the pulmonary trunk. Number four is showing you just the left pulmonary veins coming into number five, the left atrium. So we're looking at the left atrium with number five. And number six is again showing you a remnant of a fetal structure. It's showing you that soft indented region that used to be a hole between the right and left atria. It is showing you, representing, I should say, the foramen ovale, and the adult structure is the fossa ovalis. Item number 7 is showing you the left AV valve, also known as the bicuspid, also known as the mitral valve. Way up in the ceiling of the left ventricle, shown here as number 8, we're seeing one of the 3 cusps of the aortic semilunar valve. Item 13 is again showing you kind of that corrugated look of the trabeculae carnii. These are just large cardiocytes, very macroscopic structures. I actually kind of think of it, I mean, we just finished October and Halloween is my favorite. One of my favorite scary movies, only the first one, was Nightmare on Elm Street with Freddy Krueger, of course, being the bad guy. And he was horrifically burned. And I just, whenever I see his face, all I can think about is Trabeculae carnii. I know that's stupid, but it's just the weird association that my brain makes. Item number 14 is showing you the myocardium. So again, pay attention to the questions. If we tagged, just like number 14, and we said, name this layer of the heart, you should select myocardium. Item 12 is showing you the interventricular septum. Item number 9 is showing you the left ventricle. It's just showing you the chamber. Items 10 and 11 are showing you specialized cardiocytes, part of the conducting system of the heart. Specifically, number 10 is showing you the left bundle branch. And item 11 is showing you the Purkinje fibers. And again, I'm recording this, so if you need to hear this again, I don't want to write it all down because that's part of having these numbered so that you can create a slide, sort of like a flashcard, and test yourself. And then here is another heart model, and it's just filleted open for you. Someone asked earlier the perspective of the eye. So what's this perspective? It's as if the person's heart is in front of you and we're cutting it from the apex all the way up to the base. We're separating anterior from posterior. We're opening up the heart like it's a clamshell. And now we've taken off the anterior part, and this is you now looking at the back part of this person's heart. And again, we can see the atria with the little dog ears, as I like to call them, with the auricles. And inside those striated lines that you're seeing would be the pectinate muscles. And again, we can see papillary muscles. And we can see, and by the way, you need to put this in perspective. we will frequently ask you which chamber it is. So you need to know your left from your right. And here's a rule of thumb. Always, always find the IV septum first. Find the IV septum first. Then compare the thickness of the lateral ventricular walls. Which side is normally thicker than the other? The left side. So if you find the thicker ventricular wall, you know you're on the left. And the thinner one, you know that's the right side of the heart. That's important because then you know which chambers you're looking at, and also, even more importantly, then you know which valves should be found on that side of the heart. Right-sided heart valves would be the right AV valve, also known as the tricuspid, and the pulmonary semilunar valve. Left-sided valves would be the left AV valve, also known as the bicuspid, also known as the mitral valve. And also we would find the aortic semilunar valve. So whenever you look at a heart, either a picture of a model or an actual heart that's been dissected, always find that IV septum first. Then compare the lateral walls. Which side is thicker? Then that's, if it's thicker, it's the left side. And now you're dealing with left-sided structures. Is it the thinner wall? Then you're dealing with right-sided structures. This region of the heart, the pointy region, is the apex. The widest portion of the heart would be the base. Again, just identifying some of the structures or concepts that you were tested on back in lecture unit 2. Okay, now we're actually getting to sheep heart dissection. And you can really appreciate my caution to you all. Always find the IV septum first, then compare the thickness of the ventricle walls. This one's thinner, so this would be the right side, and this one's thicker, so that would be the left. Oops, oops, oops. Left. Okay, and then that makes it easy. If you know you're on the right side versus left side, then you also know which valves you could be potentially asked about. And then the next thing you should do, if you think that the arrow is pointing to a valve, then ask yourself, are there strings attached to that valve? Because only the AV valves have the chordae tendineae attached to them, and in turn attached to the papillary muscles. And here we can see this finger-like muscle that has the chordae tendineae attached to it. That's a papillary muscle. Over here is another, look at how large, it looks almost like a thumb. That's another papillary muscle. We can see the chordae tendineae attached to it. Here's my IV septum. Here is a lateral ventricular wall, and here's another one. This one is most certainly thicker, so this would be the left, and this would be right. And they've cut through the right ventricle. Why? It's actually a dissection that we do at Saddleback. We just have students fillet open that right ventricle so they can actually see the trabeculae carneae in there. This structure, it's not on your hit list. Can you see it? It almost looks like there's a worm in this sheep's heart, and it's not. That's called the moderator band. And it's not in everybody's heart. Some of you have it, some of you don't. Some sheep have it, some sheep don't. It's actually a band of specialized cells. So it's part of the conductive system. And it's basically, it's a piece of the right bundle branch that kind of cuts, instead of going down to the apex and then around the lateral ventricular walls, it's a part of the right bundle branch that just kind of grows straight across to the right ventricular wall to get it to depolarize a little bit faster. Again, not everyone has it. There's nothing good about it. There's nothing bad about it. It's just kind of neutral. Okay, and then here again, we have filleted open the heart. We've cut from the apex up to the base. We've opened up the heart like it's a clamshell, and we see the two halves here. Same rule of thumb, always look for the IV septum. Then look at the lateral walls. Which side is thicker? So this is left, this is right, and this is the IV septum. Down here is the apex. Here we see papillary muscles. We see chordae tendineae. We see a valve here attached to the chordae tendineae. Again, here's my IV septum. This wall is thicker, so this is left. This must be the left AV valve. There go all my geese. And then up here, we see another valve, just a little part of it, way up here at the roof of the left ventricle, way up here. So that is your aortic semilunar valve. We cannot see in this picture the pulmonary semilunar valve. That's part of the reason why we have students fillet open that right ventricle so they can open it up and see that pulmonary semilunar valve. But in this picture, that cut has not been done, so we cannot see it. But we can see the aortic semilunar valve. This is another demonstration on how you should find the IV septum. And always compare the thickness, look how thick the left is compared to the right. And then that helps us determine which side of the heart we're looking at. Again, we can see papillary muscles. We can see chordae tendineae. This is the left AV valve. And way up here, here's one little cusp. Here's another little cusp. Up here, we're seeing the aortic semilunar valve. Again, the layer of the heart here, this is IV septum. This would be myocardium, right? It's the thickest layer of the heart. The valves in the internal part of the chambers are lined with endocardium. And let's see. So this pin right here is tagging into these finger-like muscles called papillary muscles. Here is the IV septum, and the woman's thumb over here is on the right ventricular wall. And over here would be the left ventricular wall. Let's see, what else? Up here, you're seeing just portions of the aortic semilunar valve. And over on this side, here is a more up close picture of the aortic semilunar valve right here. And this valve right here with the strings attached to it is the bicuspid valve, also known as the left AV valve. And above it, superior to it, is the left atrium. And these muscles that you're seeing here, those are pectinate. Again, pectinate muscles are found in the atria, Trabeculae muscles are found in the ventricles. Last picture of the heart. Again, we're filleting open one of the chambers. Specifically, we're filleting open the right ventricle. I know that's hard to determine, especially because we're not seeing the landmarks that I just told you to look for. But you'll have to trust me. Look how thin this wall is. And again, I can see that moderator band that I was just telling you about. So that's another helpful clue. And all of this corrugated looking, Freddy Krueger looking muscle is your trabeculae carnii. I can see the chordae tendineae of this valve, and that would be the right AV valve, also known as the tricuspid. And here it is again on that side. And the reason why we have this picture is, again, look up here. Here's one little cusp, here's another cusp, and here's the third. So this is showing you the right ventricle filleted open to show you the pulmonary semilunar valve. And here it is again. Here's one cusp, here's another, and there's the third. It's been filleted open, splayed open like clams, so you can see that pulmonary semilunar valve. Okay, also on your hit list are some nerves, spinal nerves. Specifically, you're asked about the brachial plexus. The brachial plexus is a collection of anterior primary rami that are coming off of regions in your neck region to serve your arm. And some of these branches are going to be called the axillary nerve, for example. And again, you were tested on this for lecture unit 3, radial nerve, median nerve, ulnar nerve. And I asked you what would happen if those nerves were damaged. For example, the axillary nerve, if damaged, you can get crutch paralysis, especially if someone's crutches are impinging that nerve. Now, Lauren, we won't ask you questions like that on the lab exam. We will not ask you what kind of damage would you see in your patient if this nerve were cut.
Okay.
No. Okay.
I see that. Okay.
Okay. So we're just going to ask you. So if I tag this region, like where this bracket is, you would say brachial plexus. And if I tag this region with a bracket, you would say cervical plexus.
All right.
And the branch of the cervical plexus that you should know of, that I'll highlight in green, comes off of C3, C4, and C5. Now, technically, C5 is technically not part of the cervical plexus, but parts of the phrenic nerve, shown here in green, are from the cervical plexus. So we want you to know about that phrenic nerve. From the brachial plexus, we can see down here by the pinky, of course, pinky is ulnar side. We see the ulnar nerve, and in the middle, think M for middle, think M for median. And then by the thumb side, we see the radial nerve. Down here, we see this bracket here. If I tag these structures, you would say lumbar plexus. And the one branch of the lumbar plexus we want you to know is the femoral nerve. And the femoral nerve, I'll remind you, you've seen it quite a bit actually in other demonstrations. It travels right with the femoral artery and femoral vein. In fact, when you were learning in lab unit 2 about the pectineus, I said the pectineus is running right next to this red, blue, and yellow structure. And way back then, I was saying, here is the femoral artery, here's the femoral nerve, and right next to it in yellow is the femoral artery, femoral vein, and yellow femoral nerve. Then this bracket here is showing you the sacral plexus. And this should be ringing a bell because you just had lecture unit 3 test. And the one branch I asked you to know for the sacral plexus is this very large nerve that travels on the posterior. It's actually running through your hamstring. So it's on the posterior side of your thigh, and that's your sciatic nerve. It's huge, absolutely huge. In cats, it's about the size of your pinky. And when students pick it up with their tweezers, when they're first dissecting the hamstring muscles, they pick up this big yellow thing, kind of creamy. It kind of looks creamy, more creamy, less yellow. They pick it up with their tweezers and they're like, Kara, what's this? That's your sciatic nerve. And in you, it's about the thickness of a broomstick. Huge nerve, huge collection of axons. All right, and then going to your cranial nerves and what we can tag for cranial nerves. So unfortunately, we're not together and we're not able to dissect the cat. I love cat dissections, absolutely love it. And when we dissect the common carotid artery, it's one of my favorite dissections to do for blood vessels. Wrapped up in the tunica externa, wrapped up with that connective tissue around the carotid artery, is this very pale, creamy line that travels with this bright red artery, and that is cranial nerve number 10. It travels with the carotid artery. So here we can see the vagus nerve, cranial nerve number 10. And I'll remind you, the carotid artery is right next to the jugular vein. So here is your carotid artery. Okay, here's your carotid, specifically, that's your external carotid artery running next to your jugular vein here. And then that white line that I just highlighted in purple is your vagus nerve. Over here, spinal accessory nerve, and you can see it going towards the trapezius, and cut away from this is the sternocleidomastoid muscle. It's cut away so we can see these structures deep to it. But spinal accessory nerve is cranial nerve number 11. Spinal accessory nerve. Some textbooks just call it the accessory nerve. Cervical plexus, we would not tag it on this model. Rather, cervical plexus, if tagged, again, we would use the brackets and we would use this model. And then what else do I want to show you? The phrenic nerve. We could, I guess, tag it on this model. Here's the phrenic nerve. However, it's always been my favorite to tag it on this model, the Flatman nervous system model, because I think it's better when the student can see the cervical plexus and see a long nerve coming off of that, heading towards the diaphragm. That's another good dissection that I always like, is showing students the phrenic nerve, because they find the diaphragm when they're dissecting muscles for lab unit 2, and they see these white structures, or almost bright white structures just piercing the diaphragm. It almost looks like the diaphragm has suspenders. And students, they think it's just fat or something, and they try and pluck it off, and they can't. Nerves are very strong structures. They're very resilient. And they say, Kara, what is this? I'm like, that's the phrenic nerve. And you can see it piercing straight into the diaphragm. Okay, what about your other cranial nerves that could be tagged on models? The other cranial nerves that could be tagged on models, in addition to the vagus nerve, spinal accessory nerve, so cranial nerve 10 and 11, I just showed you. You have already seen models of the eye. That's cranial nerve 2, the optic nerve. That can be tagged on a model, and we've seen that on when I showed you the eyes. What about the ear? That would be cranial nerve number 8, the vestibular cochlear nerve. Cranial nerve 1. Cranial nerve 1 is not tagged in this lab unit. It appears better when we get to lab unit 4, when we study the respiratory system for lab. Now, we have already studied respiratory system in lecture, but in the lab, we have a lot of great half head models, and you can see cranial nerve 1 with the nerve endings going through the olfactory foramina. So we kind of go after cranial nerve 1, olfactory, in lab unit 4 when you're studying the respiratory system anyway. Oculomotor nerve, trochlear nerve, we don't have models that show those nerves, so they are not tagged. But what do? They do innervate extrinsic eye muscles, right? Trochlear nerve goes after the superior oblique. Trigeminal nerve, better tagged, again, like cranial nerve 1 in lab unit 4, because this is when you learn about the digestive system and you learn about the mouth, which is part of the digestive system when it's an accessory area, but your teeth are accessory organs for the digestive system. And we have models, half head models, where you can see cranial nerve 5 innervating your teeth. Cranial nerve 6, we do not have a model for that, but again, it's one of the nerves that innervates the extrinsic eye muscles, only the lateral rectus, which is why cranial nerve 6 is called the abducens, because the lateral rectus abducts your eyes. Facial nerve, we do not have a model that shows you, actually I take that back, we do. But again, it's in lab unit 4. We do have a model that shows the salivary glands, specifically the parotid gland. And in the model that I'm thinking about, you can see cranial nerve 7 coming out from underneath of it. Cranial nerve 9, nope. We don't have a model that shows cranial nerve 9, so you don't need to worry about it. But you should still know that it is part of the parasympathetic division. And again, you've already studied these cranial nerves. You should not have to do some heavy lifting when you're reviewing your cranial nerves. That heavy lifting for memorizing and knowing if they're sensory or motor or mixed, you should have already done that heavy studying for lecture 3. Cranial nerve 10, the vagus, I just showed you on a model where we could tag it. Accessory, spinal accessory, cranial nerve 11, I showed you where we could tag it. Cranial nerve 12, hypoglossal, the tongue, again, better tagged in lab unit 4 when we're studying the mouth, accessory organs like the teeth, the tongue. We have a model showing the nerves, cranial nerve 12, going to your tongue. So Lauren, did I address your concerns now?
I think you did, but I also had another question. Is the sheep brain going to also be on the exam?
Yes.
Okay.
And we did cover that.
Oh, right. That's right. No. I got confused. I blanked out for a minute.
Yes. So eye, ear, brain, blood vessels, histology, heart, nerves, that's all lab unit 3. Absolutely.
Okay.
Now,
Thank you.
I have a short practice lab exam for you all. It's only 40 questions. that gives you a very good sample of what to expect. I have questions on there, just a few questions: eye, ear, models of heart, sheep heart, models of brain, sheep brain. When I say eye, we have pictures of eye models and also sheep eye dissection. I have a good assortment of blood vessels that are tagged. Anyway, I have allowed you to take it 5 times over, and it will show you the correct answers. Again, as usual, if you find a discrepancy, let me know. The part of that practice exam and official lab exam, so myself and Dr. Lopez, we kind of shared the responsibilities. So if you find something that is a discrepancy or you're concerned about, you should let me know sooner versus later, so I have time to address it, fix it if need be. Anyway, it's only 40 questions. You can take it 5 times over, and you get 5 points extra credit towards your lab exam for taking this. So if you choose not to take this practice exam, you don't have to, but you don't get those 5 extra credit points. It will not count towards any other points. So it's not counted as a quiz. It doesn't count into your official grade. This is just for practice to have an idea what to expect for your lab exam 3. Are there any questions? Have you looked over your lab hit list like I have? Do you have any concerns? Something that you think I have not addressed? Because I went through it today and I feel like between me addressing things and your lecture unit 2 exam and lecture 3 exam that you're well served. But you tell me any questions you have. Okay. Well, I will stop recording this.